smash the like for this next upcoming presentation. I'm excited to learn a little bit more about ContraFect. It's ticker CFRX on the NASDAQ. And there is the chairman and CEO, Roger Pomerantz. Roger, how are you today?
Very well. Good to be with you.
Thank you very much. It's good to have you here. It looks like you have a nice office space wherever you are. It's got to be in your upstairs, right?
It is. It's in New Hope, Pennsylvania, where we have a farm.
Awesome. Well, like I said, I'm excited to learn about ContraFect. I'll let you take it away. I see your slides. Looks like we're good to go.
Great. And thank you for that. It's great to be here. I am Roger Pomerantz. I'm an infectious disease physician scientist by training. Just real brief, so those who don't know me, I left academic medicine and went to the dark side to run Tibotec, which was a company that made antiviral drugs. We sold it to Johnson & Johnson. I stayed there as global head of ID, was also global head of ID and business development at Merck, started Series Therapeutics, and then came out of retirement to lead ContraFect, whose board I was on for some time. Let me tell you why. As an infectious disease physician, the last 10 years have been terrible in antibacterials. There has literally not been one drug that moved the needle. It's been the worst time in antibacterial antibiotic development since the 1930s when sulfa was developed. And it became clear that just banging our heads at getting at new antibiotics, which are small molecules that work on the bacteria inside and their RNA and DNA and enzymes, was not working. So ContraFect came up with a new modality approach. Next slide, please. Next slide. We are doing what has been done in other problematic areas in biomedicine. People know about gene therapy. Rare diseases were never tackled before that paradigm shift. In cancer, before immunotherapy using PD-1, there was just chemotherapy. That revolutionized that group. We feel that in anti-infectives, we need to think past just basic antibiotics to something that's completely new. And we believe we have that. They are called direct lytic agents. They are not small molecules. They're enzymes or peptides. They come out of bacteriophages. They are not affected by resistance to other antibiotics. And we believe we also know the trifecta of how you make a drug in the antibacterial space that people need, want, and will use and will be paid for. You need superiority, not non-inferiority to standard of care. If this is a highly mortal disease you're treating, you have to change mortality. You have to save lives. And for payers, you have to put money back into the payers' pockets using positive health economic data from your slides. If you get those three, we believe that is the thesis that will change what has been a lagging field. Next slide, please. As I said, we have a number of different direct lytic agents. Remember that name. The first and the farthest along is Exebacase. It is a direct lytic agent, an enzyme called the lysine that has gone through phase 1, phase 2, and is in phase 3 trial right now. What's interesting about this drug is that for the first time in 50 years, we've been able to show a difference in patients that have Staph aureus bacteremia, especially methicillin-resistant Staph aureus, MRSA, which is the worst of the Staph strains. And what we showed is something that was really a remarkable data set, that we could improve standard of care by almost 50% against MRSA. When people saw this, the FDA gave us breakthrough therapy designation, which has been very rare in antibacterials, fast track, as well as streamlined development, meaning we will only need one phase 3 with compelling data, not two. This is the first investigational non-antibiotic anti-infective developed. And as I said, we're in our phase 3 superiority trial. Remember, as I said, we are not looking for non-inferiority. We need to change the paradigm, like in those other therapeutic areas. But we're not a one and done company. We've also developed license that get gram-negative bacteria, some of the worst bacteria and infections that take place in surgical suites and oncology areas. And we have a drug, CF370, that gets Pseudomonas aeruginosa. Pseudomonas is something that many people know about. It kills many people around the world each year. And we have a drug targeting that. And then on top of that, we have certain peptides called amurins that are broader, that get a large number of bacteria, in particular gram-negatives. They get all of the escape mutants, ESKAPE, which are the worst gram-negatives according to the Infectious Disease Society. Next slide, please. Next slide, please. Yeah, and it just is important to see. I like the history of medicine, and penicillin was developed for staph in the 40s. It got everything until resistance developed. Then vancomycin came in 1958, and there hasn't been a change in therapy that's better than vanco since 1958. That's a very long time. This is not a rare disease. It is a neglected disease. Another drug was developed in 2006, daptomycin, but that was just non-inferior and therefore gets very little of the market. Exebacase was the first data set that in phase 2 that ever showed superiority to these drugs in over half a century. Next slide, please. MRSA, everyone's heard about it. It's not only there, but it's getting worse. It's increasing in the U.S. The USA 300 strain has developed here, which is much worse than the original strain. Patients have been getting it with other comorbidities, and it shows when you have USA 300, even more of the metastatic horrible infections. What's important, again, to remember about Exebacase is it doesn't matter about the strain. It will get all Staph aureus. Next slide, please. The key attributes of this, I've talked about. It's an enzyme. It works very fast, in seconds to minutes instead of hours to days like antibiotics. The clinical stage program, the Disrupt trial, is moving forward in over 60 centers around the country. We are battling through COVID because people still get staph even through the COVID epidemics, and we are gathering patients in new sites all the time. Next slide, please. What's really important as well is when you think about what a drug can do, you look at phase 2. And when you look at phase 2 for MRSA patients or all staph patients, meaning every staph, not just MRSA, we were able to show truly profound differences in phase 2, which is why I think we got breakthrough and all the bells and whistles from the FDA. We also got a grant from BARDA, from the US government, for almost $90 million because they thought it was this important. Next slide, please. This is something that is a new piece of data from phase 2. We not only showed better response, a change in mortality, and health economics that makes it worthwhile for payers to approve and suggest that it can be priced well. But we also saw some new data when we looked at phase 2 again. If you look at how symptoms and signs of the disease go away, they disappear in half the time if you add Exebacase to standard of care. And as a clinician, this is how you would think about these patients in the hospital. Are they getting better quickly? Because the die is cast in this disease early. We were very pleased with seeing this data. Next slide. Again, we are moving a single pivotal trial forward. It's a very simple 350 patient trial, randomized 2-to-1, active to placebo. It's in the US only, but this is a global drug, and we have talked to our friends in Europe. We're moving this forward. We expect to have the futility analysis in the first half of 2022. And what's important is that this is a superiority futility analysis. We don't want to make a non-inferior drug. If it ever fell below superiority, we would kill it. After that, we expect sometime in the end of 2022 that we will see the full data set. This is what we expect to see. Additional design, we have powered all the endpoints, not just the primary endpoint, but mortality, not just MRSA, but all staff, so that we could possibly get a label depending on the data, but that would allow us to look at all Staph aureus complicated bacteremia in the United States, and that would be another huge success. Next slide, please. Exebacase value proposition, it meaningfully in phase 2, and as we hope and expect in phase 3, it changes the primary outcomes in patients with MRSA bacteremia. It would change mortality, as we've seen in phase 2, and expect and hope to replicate in phase 3. And it also has a true effect in the reduction of length of stay in the hospital, and 30-day readmission rates, both of which are important when you talk to payers, as I've done many times in my career. MRSA bacteremia also makes something called a biofilm, which hurts antibiotics. What's interesting about DLAs, direct lytic agents, is that they lyse biofilms. Next slide, please. If you look at the cost of these patients, they're the sickest and really some of the most expensive patients in the hospital. I've taken care of, in my academic career, hundreds of these people. And what you see, and this is 2013 data, it's more expensive now, that if you have MRSA bacteremia, complicated bacteremia, your spend is close to $115,000 with a range that goes out beyond $250,000. Even methicillin-sensitive staph is almost $50,000. And this does not take into account the many readmissions that take place soon after discharge. Next slide, please. What was really interesting is we saw such important data in phase 2 that we asked, could we treat other staph infections that are high unmet medical needs? And so we thought about prosthetic joint infections. Very important, difficult to treat. Again, something that I treated many times. There has been no change in therapy here for the last 50 years, and the outcomes are not good. But what we did is show with a compassionate use trial, now with almost 20 patients, that if you inject Exebacase into the infected joint, as you see in some of those pictures, fistulas close, people get out of bed and have less pain. This is anecdotal evidence because it is compassionate use, but when you see changes like this, you expect that this drug is likely having an important effect. Expect us to start a phase 1b/2, a proper trial with this drug injected directly into the prosthetic joints that are infected with staph. Next slide, please. Again, next slide, I've talked to you about CF370. This is for gram negatives. It used to be said that you could not get into gram negatives with a license, with a DLA. We were able to molecular engineer this at ContraFect, so they enter the gram negative bacteria through what is called the outer membrane, the large hydrophobic wall, which makes gram negatives different than gram positives. And once again, just like Exebacase, once we got them into the bacteria, they did their job rapidly, and targeting bactericidal activity, eradicated their biofilms, worked in seconds to minutes instead of hours to days, again, different from antibiotics. Next slide, please. And we showed it as well in a Pseudomonas lung infection model where we saw an effect and then synergy with antibiotics, standard antibiotics, where we lower the bacterial cell count in these lungs down orders of magnitude. This is very exciting, and this is what we are going to be moving to IND and into patients likely with hospital-acquired pneumonia with Pseudomonas. Next slide, please. I should have also pointed out we even made another hard model. We tried to stress these new drugs that are these new modalities. And so we used an infectious endocarditis model in rabbits, one of the hardest to treat of Pseudomonas infections, common in IV drug abusers. And once again, the drug performed remarkably with orders of magnitude decreases in Pseudomonas in the heart valve, but also in organs around the body, since these animals are bacteremic, just like humans with endocarditis are. Next slide, please. Again, when you look at CF370 and gram-negative pathogens, it's not just for Pseudomonas. It was developed for Pseudomonas, but it has activity in other bugs. This is also very important as we think about how it could be used. We even showed that it has an effect on the very difficult to treat mucoid Pseudomonas that are in cystic fibrosis patients. Next slide, please. Amurins as the next part of our pipeline are active against gram-negatives. They are peptides, but not enzymes. They do come out of bacteriophages. And once again, they have remarkable effects on a wide variety of gram-negatives. These are some of the best antibiograms I've ever seen for these infections. And what we like about this is that infectious disease is not just treating one bug with one drug. Most ID docs treat patients with a broad spectrum of antibiotics, and they design a regimen. That's what we would like to do in the future, is design a regimens using DLAs rather than antibiotics, because that would really change the field as we know it. Next slide, please. So again, we've talked about the investment highlights. We have Exebacase, which we expect to read out next year in its phase 3 Disrupt trial. We expect to have 370 going through IND and into patients for Pseudomonas, and then we'll bring Amurins forward for the next IND. I should note that besides all the things we have, you'll note on this slide, Pfizer. Our friends at Pfizer have been multiple equity investors in our company. They also have their head of hospital medicine on our scientific advisory board of ContraFect and help us in kind with manufacturing and such. We've been very happy with our collaborations with Pfizer. I should point out, though, that they do not have rights in the company. All of this is wholly owned at this point by ContraFect. Next slide, please. Again, these are some of the particulars of the financial information. I'll end the call there and wonder if there are any questions.
Here I am, Roger. I have a couple quick questions, if you don't mind. So, wanted to ask a little bit more about Exebacase. So, I'm sure there's a lot of people in the audience who are familiar with FDA process and biotech companies process, but you said, I think that you were expecting phase 3 interim results in the first half of 2022. Is that correct?
Correct.
So, and then you said next is a label, but I just wanted to, break it down for some of our audience who might not understand this stuff as well. I mean, what happens after the label?
Yeah, so I've done this a few times with other drugs. As I said, I have a 13 that I've led the development of, and I used to be on the antiviral advisory panel, so I know the FDA pretty well. What happens is it depends on what you have as the nomenclature from them. With breakthrough, sure, we get it done in 8 months versus 10 months. If there's an advisory panel, sure, I like advisory panels. That's like your first commercial. That's what Wolf Blitzer will talk about on CNN. So I want an advisory panel. But what's good with breakthrough is it does not hit your critical path. It does not delay you. They have to put it in within that 8 months. So it's a relatively rapid, as these things go, path to advisory panel and hopefully approval.
Gotcha. And then I just wanted to ask a question. I'm not sure if you were speaking broadly or if you were speaking about that last segment that you were just focused on in your presentation, but you said, if we could change this to be more like a regime.
Regimen.
A regimen, excuse me. So, I mean, dive into that really quickly a little bit more. I mean, what would be the advantage?
The advantage is if you have an advantage on getting past resistance for one drug, one bug, and you can do this broadly. Let me give an example. Most AID docs, like I am, when we see patients, we have to cover them broadly because many times infections are polymicrobial. When that happens, if we can use DLAs instead of antibiotics and develop a regimens or different regimens for different indications that are polymicrobial, then you get by the resistance of all the bugs that might be in there.
Gotcha.
That's what you want. You want to, this is not your father's antibiotic. We do not want to treat one easy to treat infection like a cellulitis.
Right.
We are looking like it's a high bar, but we're looking like PD-L1, PD-1 for oncology that changed the way it looks. We're looking at it like gene therapy, which made rare disease now a path to true therapy. I usually start these talks with saying we are not an antibiotic company. You can believe me, I've spent my life with antibiotics. This, if we are successful as we expect, will change how you look at treating bacterial infections.
Love it. Well, Doctor, the response in our chat on YouTube was extremely optimistic. I mean, you got some fans out there. People are impressed with the technology and what you're doing, and extremely commendable thing that you and your team are working on. I love that you said, you came out of retirement because it seems like you're extremely passionate about this, and obviously something that you want to focus on. And it seems like you're absolutely trying to change the world.
Well, we would hope so. I think the world in antibacterials needs changing.
Sure.
Like I said, I was head of global business development for Merck. I'm an ID doc, and I never bought an ID company. And that was because there was nothing that really rose to the level that Merck would want. What we want to do is show that we don't have to keep doing the same failed experiment over and over again. Keep throwing E2 antibiotics. So that's what we'd like to see.
Well, thank you so much for joining us today, Dr. Roger Pomerantz. He's from ContraFect. It's ticker CFRX, and you are the chairman and CEO. Thanks for joining us. Have a great rest of your night. Really appreciate it.
You take care. It's great being with you.